Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It is our great pleasure to have Dr. William Taylor Kimberly. Dr. William Taylor Kimberly is the Chief of the Division of Neurocritical Care at Massachusetts General Hospital and Associate Professor of Neurology at Harvard Medical School. He is a neurologist who cares for patients in neurocritical care ICU and in the outpatient neuro recovery clinic. Taylor leads a research group that conducts transnational studies, bridging basic science and patient-oriented research. He's recognized for developing blood and imaging biomarkers to inform phase 2 and 3 clinical trials in brain edema and was the PI for several national and global randomized clinical trials. His research group also investigates the relationship between diet and the gut microbiome and the risk of stroke and dementia. His laboratory has also studied clinical application of low-field MRI in the inpatient and outpatient neurologic settings. I'd like to remind our audience to address their question at the end of the talk. Without further ado, the stage is yours. Thanks. Thank you. Appreciate that.
Well, it is a privilege to be here. Thank you for the invitation and the opportunity to talk with all of you. Hopefully, spend a little time afterwards meeting you. And today, I'm going to focus the talk on really what has been a highlight of my faculty career, which is a multidisciplinary collaboration around portable low-field MR. I want to start out by acknowledging some of the team members and collaborators. This by no means is a comprehensive list, but I did want to acknowledge right at the outset that some of the work I'm going to be presenting is really the product of a broad multidisciplinary team where each person brings a different set of expertise and we all coalesce around what we hope is an important clinical problem, and bringing the emerging technology of low-field MR to that. On the left are some members of my group starting back when we first began scanning now over 5 years ago. And I do want to highlight in particular Annabelle Sorby-Adams, who really is a senior postdoctoral fellow in the group who has been an incredibly important leader on my team, as well as Stephen Bakke, Ava Farnan, and Amelia Demopoulos, all of whom are actively enrolling, scanning in patients, and then analyzing results. The wonderful part about this has been the broad collaboration, and you'll obviously recognize many of the faces in this group back now, gosh, like 8 years ago, Matt. And when we first started on this journey, you had already co-founded the company, and connecting up with a very good collaborator and friend of mine, Kevin Sheth, in the upper left on that right set of headshots. It really started a journey that I've found incredibly enjoyable and scientifically and clinically very gratifying. Also want to acknowledge John Kirsch, who many of you know quite well, and of course Eugenio, who has been a fantastic collaborator now for 4 or 5 years. And then finally, Teresa Gomez-Isla, who is the chief of the division of the Memory Disorders Unit, now Mass General Brigham Wide. And she has been a great collaborator for some of the work around Alzheimer's disease and memory disorders that I'm going to share here today. So what I'll do is I'll just give a brief background and history. Honestly, some of you can provide the background in a more informed way, so please bear with me and cut me a little slack as I try to explain the background, but also some of the history that got us here today. And then the main focus of today is really talk about how, as a clinician scientist, how we think about working together with many of you here in the Martinos Center, how to bring it towards use case scenarios. And I want to show you some of the nuances, challenges, but also the opportunities, and I'll focus on two examples, and then of course future directions. I know questions are supposed to be saved at the end, but if you really have a question that comes to mind as I'm talking about something, I certainly don't mind if you just raise your hand or belt the question out. I am happy to answer it in real time. Okay, so this everyone here will be quite familiar with, but one of the exciting things, at least from my perspective, is some of the unique differences between low field or ultra low field MR, depending on who you ask, and conventional MR, which when I mention that term, I'm really typically thinking of 1.5 to 3T because that's really what we see in the clinical realm most frequently. I won't belabor the differences except to say when you have a permanent magnet with no cryogens and you have active electromagnetic noise cancellation, you suddenly can unlock portability. And that is, as a clinician, incredibly exciting because all of the paradigms that we think about for how to use MR imaging in clinical care suddenly are turned on its head, and it creates new opportunities. There are, as we all know, but certainly something we don't experience day to day here at MGH or in an academic medical center, but it turns out actually there's a number of very substantial barriers to the use of MRI. As we all know, it's pretty costly to install a 3T scanner in a clinical site because of all of the site requirements around shielding and the weight of the instrument, the capital investment itself, and then, of course, all the maintenance costs related to cryogens and electricity. And that really limits, you basically have to bring the patient to the site, and that limits, just by virtue of that geographic restriction, limits or creates a barrier. And because of those costs, as we've all seen, there are many ways in which that can contribute to inequalities in access, and that's both locally as well as globally. And that obviously interacts with patient factors that as we see patients where we want to get an MR scan, oftentimes there'll be limitations that some of which we may not even be aware of. And having to set up a separate appointment where a patient goes to a specific location to get a scan, it sounds easy to describe, but it actually ends up being a substantial barrier. And that doesn't even account for the cost in the form of co-pays or insurance coverage or lack thereof when we're trying to get these types of imaging. So if you think about access to MRI on a global scale, moving beyond the Boston area where we are heavily resourced when it comes to imaging, and we're really very fortunate in that respect. But when you think about it globally, there's actually wide differences in the number of MR scanners per million people. And even in the regions of the world that are dark green, which have the highest density of MR scanners by country, take the US, for example, there's really wide regional and local variation in access. So obviously there's a lot more granular detail to this broad story that access is not uniformly available when it comes to MR, and by some reports, a vast majority of the population of the world doesn't have regular access to MR. So as I mentioned earlier, portable MR or making a low field MR scanner portable suddenly creates new opportunities in thinking about imaging and neuroimaging in particular, which is, as a neurologist, the area that I'm most interested in. And we all know the current paradigm where we have to bring the patient to the scanner, and all of the requirements, safety, screening, and otherwise that come along with it. But when you realize that think about the fact that the low field scanner can be brought to the bedside, there are opportunities where you can bring it right to the point of care. And we'll talk about two examples in just a moment. But there are other ways in which the portable low field MR can be deployed. and that could be out in the community, either in the pre-hospital setting. You could put it in an ambulance. And for example, mobile stroke units are currently available that offer CT scanning. Now mobile stroke units with MR is certainly a possibility, as well as in community health centers, and then also improving access in rural, low resource areas or low and middle income countries. All of these are on the table as far as I'm concerned from a clinical implementation perspective. But one of the reasons that we focused on that top set at the point of care is really because of the logistics of MGH as an academic medical center, but it's also a recognition that there are a lot of challenges to solve to make the technology and the systems robust enough to reliably be deployed in lower resourced, lower access systems. So this is where we have the opportunity to define how this technology can be deployed, develop tools and technologies to make it robust. And then the hope, of course, is that then that can be deployed in other settings where there isn't a center with the world's leading MR and imaging researchers right next to a hospital, where they're really interested in just a deployment for clinical purposes. So the places where we are routinely scanning currently, I'm a neurologist and neurointensivist by clinical training. So actually the first place we started was in the neurocritical care ICU back during COVID. And it was kind of a risky place to start, but both Kevin Sheth and I are neurointensivists. That's our home base clinically. And we had a lot of trust and partnership with the nurses and the nursing staff. And so we have a lot of experience in the ICU. We've also done it in the emergency department. And now increasingly, as we explore new use cases, we have a location in the neurology outpatient clinic, which you can see on the right-hand side. So just briefly, a little bit of history about the development of this technology, because one of the amazing things about it is, at least from my perspective, I feel like I'm holding on for the ride because things are changing so rapidly. It's really quite fun. And so this slide is really an attempt to capture the rate of change in this field. So back in, I don't know, Matt, was it 2015 when you co-founded the first commercial company? Maybe it was 2016, 2014. So that was before my time. I got connected up with Matt and Kevin Sheth back around 2016 or so. And around that time, the first image was coming off a commercialized system. There, obviously, I should mention, there are many low field systems that are in the research space right now. There is one commercial system, and I'll share data and images from that, but I'm enthusiastic about the prospect of additional commercial or manufactured systems at various development stages, but hopefully will contribute to the clinical imaging space of low field imaging. Anyway, in 2016, this was the first image that was coming off of a human brain. And when Matt co-founded Hyperfine, they set up shop down right outside of New Haven in Guilford, and partnered again with Kevin, who is a neurointensivist at Yale. And so they got the first system and they brought it into their ICU. And this was the first patient that they were able to stick in a scanner and get pathology. The image took 15 minutes to acquire. This was a hemorrhage, a basal ganglia hemorrhage. And it was the first pathology that was detected. This is 7 years ago. And then that rapidly followed a series of innovations. The first DWI, single direction B900 image, came out in 2020 around the time of COVID. And in fact, we started using it to image critically ill COVID patients to detect occult stroke, which was an issue around that time. And the image acquisition times were coming down. About 3 or 4 years ago, partnering with Eugenio and his team, we started to recognize that the image quality could be combined with machine learning post-processing tools. And of course, in parallel to this, there was a lot of improvements on the pulse sequence development and the image reconstruction that was happening on the company side of things. And that led to improved SNR. They also spent some of that improvement on reduced acquisition times. And now we're getting about 5 minutes per acquisition per sequence. Still a little longer than we'd like, but certainly within range from a clinical perspective. And over that period of time, we've spent some time just kicking the tires, testing it out in a variety of scenarios, trying to get experience with using this technology in different settings and with different conditions or diagnoses. And these are just some examples. On the left, ischemic stroke patient. The two image slices on the left are the low field. The one on the right is a standard of care head CT. In the middle, intracerebral hemorrhage. And then on the right, cardiac arrest. And what these studies showed, besides these proof of principle photos or images that I'm showing you, is that it really defined that it was feasible. And that was a really important question for us. I mean, we could drive this around in the hospital, which is there's obviously a lot going on, a lot of other equipment, a lot of other activities. And I'm pleased to say, and really Kevin Sheth and his team spearheaded all of this work. They really set the stage for ensuring that once we started scanning, we knew it would be safe. And we've contributed to that safety profile of this technology in a variety of settings. So now I'm going to talk about a couple use case scenarios and give you a flavor for where we're at now for the types of questions we're grappling with, some of the projects we're partnering with many of you on to try and figure out how to make this technology practice changing. And one of the sort of general comments I'd make is that there are a lot of diagnostic tools out there, and it's a big deal to show that a tool can improve or detect disease and also detect normal, but that's not enough to bring it into clinical practice. And you really need to, in that scenario, you really need to show how the technology can be folded in or influence clinical workflows. And so one of the areas that we were very interested in was stroke and figuring out how to use low field imaging for acute stroke. One of the challenges, of course, is that acute stroke treatment and the clinical algorithms and workflows are really time sensitive, and there are many decision steps along the way. So you can't just roll the scanner into the ED and start scanning stroke patients, because that will interfere with time sensitive clinical decisions. And frankly, even if you had the best imaging technology that took 5 or 10 minutes to acquire, it may not even be necessary in scenario. You might have a scientific question, but from a clinical implementation perspective, it's not relevant. And so, in showing this simplified workflow, I wanted to try and convey a flavor for that. That when a patient comes in with a suspected acute stroke or perhaps a TIA or transient ischemic attack, there are a number of decision points along the way. If it is a mild stroke, well, then they won't qualify for all the time-sensitive treatments like TNK or TPA, thrombolytic treatments, or mechanical or endovascular treatments like endovascular thrombectomy. But there are then a number of questions around triaging. Is it a stroke or is it not a stroke? And those have to occur in the emergency department where time is of the essence, given how busy ED departments are. And so the question there ends up being, is this look like an acute stroke, or is it an acute stroke where they might need to be admitted, or can they be discharged from the ED and have the evaluation and treatment plan implemented in the outpatient realm? I should say, actually, almost half of acute strokes or TIAs are mild. So we don't think about them clinically all that much because there isn't as much acute decision-making. For those that have more severe strokes, then the decisions are whether to give TPA. That's a head CT-based decision where we now have quality metrics where that CT has to be gotten within 15 minutes. 15 minutes of the ED of the patient arriving into the ED, coming through the doors. And then some of those patients, you might not know when their stroke started. And at least in the setting of thrombolytic decision-making, we make that decision on the basis of when they were last seen normal. So if someone went to bed the previous night and they woke up this morning, let's say they woke up 2 hours ago, we'd have to use that time from last night, even though evidence suggests that they might have woken up because of the onset of the stroke. So there are some ways in which MR could be used in that scenario. And then finally, now the eligibility criteria over the last few years through a variety of clinical trials have expanded the use of thrombectomy. And so a lot of the decision-making around that is CT, CTA based. So there are two areas in which, in this algorithm, which there are a series of decisions being made where MRI could be useful, or at least that's what we've been thinking. So one is in these mild stroke patients who aren't going to qualify for thrombolysis or thrombectomy. And really, it's an ED triage question. Do you have a stroke or not? Can you get an outpatient workup, or do you need an inpatient workup? So that's one area. But there you have to be able to detect small strokes, because remember, this is a mild stroke population. So that's one area. The second is these wake-up strokes. And that is where the example that we decided to first focus on as we were thinking about using a low-field MR in acute stroke. And so what I'll share is a little bit about the background about wake-up stroke. I mentioned that there are a number of patients, probably about a quarter or so of patients, do not have a witnessed onset time. Many of those go to sleep, as I mentioned in that prior example, and wake up with symptoms. And in that scenario, we typically don't give TPA or TNK because they're outside of that 4.5-hour window based on when they were last seen normal. But what investigators both in Europe as well as here, Ona Wu and Li Schwam had been studying this. The idea was that the MR imaging and FLAIR in particular could be used as a surrogate for your time. And that if your FLAIR T2 hyperintensity didn't show up, and they defined thresholds for that using a quantitative signal intensity ratio, that could be used as a surrogate for time. You could give TPA. And so this concept of detecting the stroke on DWI, but not seeing it on FLAIR, that mismatch, is a surrogate. And this I've mentioned here, it's called a tissue clock, but it's really just a time surrogate. And these examples on the right show that, and I'm not sure if I can, yeah. So in this case on the left, there's a clear DWI lesion and no FLAIR hyperintensity. That is a very early stroke. The example in the middle, there's some subtle T2 FLAIR hyperintensity. And then on the right, there's a much brighter T2 hyperintensity. And this concept was actually tested in a randomized controlled clinical trial. Gertz Tamala and colleagues in Europe ran this trial. They called it the wake-up stroke trial. And what they found is if a clinician's got a hyperacute MR, saw DWI FLAIR mismatch, randomized into regular care or regular care plus TPA, that in fact patients did better if they were treated with TPA. This graph that I'm showing you is a modified Rankin scale plot where the lower scores are better functional outcome at 3 months. It's a very typical stroke outcome measure. And in general, if you've got a bigger box that's a lighter color, that means you've recovered better. If you're a dark box, and in fact, if you're a black or a very dark blue box, you've died. So you can see that the lighter boxes with the alteplase arm are bigger than in the placebo. And this was quite statistically significant. So being able to select patients with MR to guide treatment has level 1 evidence that it improves patients' outcomes. This was in 2018. Guess what? It's very, very hard to do this. And there are very few sites that can. Why? Because no one has, or I should say very few sites. We do, but very few other sites and centers have hyperacute MR capability. So with leveraging this concept that low field MR can improve access to MR, we said, well, gosh, what if in a future state, every ED has a low field MR scanner? Could this be used to select out these patients? So we started just with proof of principle. Does the T2 flare imaging on low field behave similarly as we know it does on high field? And so that same concept is shown here. Again, 3 different examples of patients. The low field DWI is the top row, the low field flare, the bottom row. And it's vertically aligned where each patient is a pair above and below at different time points. And indeed, on some initial experience, we were encouraged by what we found. So we pulled together a multi-center group. We connected up with our good colleagues at Yale and brought in some other colleagues from Buffalo, Ohio State, and then us. And we all had a scanner sited in the emergency department. And we were all consenting and enrolling patients with acute stroke symptoms who were presenting within 24 hours. And of course, one of the challenges is that we couldn't interfere with standard of care. So it was a tour de force to pull this off, and that was through the hard work of our collaborators in the other centers, as well as Annabelle and the ED team here. And even with all that effort, we only were able to get 10 patients in that less than 4.5 hour window. We got 6 in the 4.5 to 6 hour window, and then the majority were in that later time point. And the bottom line is what this table shows, or what you have to take my word for it, is that these are all mild strokes because we couldn't interfere with the severe stroke cases that were being treated or evaluated for TPA and TNK. And that's okay, because we knew that's where if we could show value in mild strokes where the strokes are smaller, then we would have no problem with more severe strokes. And so what we did is we superimposed the DWI lesion on top of the flare images. We developed a signal intensity ratio on the flare sequence. We did not come up with that. That was Ona Wu and Li Schwam's group. But we replicated that at low field. And then we asked, what's the optimal cut point in that flare signal intensity that defines 4.5 hours or less? What you can see is that the flare signal intensity goes up as time proceeds, and developing a receiver operating characteristic curve, we found that a flare signal intensity ratio of 1.15 was the optimal cut point. And that pleasantly surprised us, because that is exactly the cut point that Ona and her collaborators had found as well. Now, maybe it's not a huge surprise. A flare is at low field, presumably has similar imaging characteristics at high field, but it was reassuring to see this, particularly if in a future trial, we're going to use this to select treatment for medication that could cause death. So in doing that work, one of the things we realized is that the acquisition for this DWI ADC was long. It was 8 to 9 minutes. It varied over time as the manufacturer improved their pulse sequence, but it was in that time range, 8 to 10 minutes. But one of the you know, one of the limitations is that it's one direction. It's one direction. So when you think about a clinical 3T MR or any of the DTI image acquisitions that are acquired here in the Martinos Center, it's 60 directions or 30 direct, 20, 21, whatever it is. It's a whole lot more in a lot less time. And that's just the reality of imaging at low field. And so in doing this, running this study, we realized that there needed to be improvements in the DWI acquisition to make this clinically useful. That if we wanted to run a trial or a study where we just relied on a low field scanner, that it wasn't quite ready for prime time. So that really got us thinking as well along the lines that that we needed to boost sensitivity, particularly with the small strokes, and also because of its potential value as a triaging tool for TIA and minor stroke. So, as I was saying earlier, the manufacturer was not sort of sitting on its on the same sequence or even the same software, not even the same hardware, and that they were actively making improvements in not solely in response, but as we were gaining experience, we engaged in a bidirectional conversation about how to, where we wanted to take this technology. And to their credit, they were responsive to those conversations, and that really allowed us to evaluate different versions of hardware and different versions of software. And so I'll show you an example of where we started, which is the top row, which was the hardware version 1.6 and an earlier version of software 8.2. And the first is obviously a 3T DWI, beautiful. You can see the stroke, no problem. You can see the middle low field single direction B900 is entirely useless, and but you could certainly see it on the low field flare. This was a subacute stroke that we were imaging. But on the lower panel of images, using a version 1.9 of the hardware, which had some improvements in the gradient coils and the amplifiers, as well as improvements in the software, which is largely centered around image reconstruction. Then you can see actually the single direction B900 in the middle is pretty good. It's pretty good relative to the gold standard, which is of course the 3T DWI, and that the flare has also demonstrated improvements. But one of the challenges of a single direction acquisition is that, of course, there is intrinsic restricted diffusion within the brain largely based on the structure of the white matter tracks, and that's obviously used to leverage to great value in the setting of DTI. But if you're trying to detect an acute stroke, and that's really a problem if you're only collecting one direction. And this was a sentinel case that really drove home this particular limitation, where you can see on the left the DWI and then the ADC. And while you can sort of see a small lesion that is likely a stroke, it is bright on the B900, but it really is entirely obscured in the ADC based on the directionality and restricted diffusion of the white matter tracks that are intrinsic to that area. Whereas the gold standard scan, it pops out very easily, and you can see it as restricted diffusion on the ADC as well. And so when we saw this, we asked, was this intrinsic to low field, or was this intrinsic to a single direction? And this is really where John Kirsch has been instrumental in thinking through these challenges. And we actually pulled out the individual directions on that high field image and of another case in a similar location. And even at high field, the signal to noise is not as obvious when you're looking at a single direction. And for us, that really highlighted that for this to become a clinically useful sequence, we need to move away from single direction acquisitions, which again, with John and Annabelle's close partnership together, they were able to develop. And this was that case using 3 direction acquisitions. We were able to trim the time as well, so it's still long, but it was about a 12-minute acquisition, not 24. So we got it only a little bit longer than the stock sequence that the manufacturer provided. And I hope you can see that both on the DWI and the ADC, there's a more homogeneous signal surrounding the stroke, making it easier to detect and increasing the confidence. We've quantified that by looking at the standard deviation in the signal intensities on both the DWI, which is the left-hand panel, and the ADC on the right-hand panel. And basically, each pair of colored boxes, the left-hand box is the single direction, the right-hand box is the 3 direction. And you can see in each case that standard deviation drops down. We looked throughout the brain in a variety of different regions, of course, excluding the stroke itself, with the idea that if we can get a more homogeneous tissue signal around any stroke, it will help clinicians and neuroradiologists in particular to detect and diagnose stroke. So, okay, so that's the multi-direction or 3-direction DWI. And I think that's all I'll say about this. So I would say that our sense at this point is that to use low field MR for stroke, we've got to have 3 directions. I was saying something that is self-evident to all of you, but that is at least empirically shown. And now the manufacturer's in the process of setting up a stock multi-direction sequence that will be rolled out more widely. I will say then that the 2 leading use case scenarios that I've talked about is in the setting of wake-up stroke, leveraging the FLAIR sequence in combination with the DWI. And then, of course, once we can validate the lower limit of detection of small embolic strokes, that's the key that would unlock using it as a triage tool in the emergency department. I think there are a variety of trial designs. I won't dwell on it except to say that my sense is that this is not like a standard double-blind RCT trial. I don't think that's the way to pull this off, but rather a pragmatic design or a comparative effectiveness design. I do think that will be necessary to drive adoption among clinical providers. I think it's not enough to say it can be done. We have to say that it is equal or better, or hopefully better.
Well, in this setting of that wake-up stroke, especially small ones, how important is the assessment of hemorrhage, absence of presence, and obviously that might seem to be more challenging in the acute setting. Obviously, you're not in the medically broken states the way or an exam ratio. So, where does that part of the scope fit in?
Yeah, Bruce, it's a great question. So
One you wish to have.
No, it's one we grapple with all the time, and it's an important one. So right now, hemorrhage detection on the low field scanner is possible in the subacute phases. Let me say, it's feasible. It's feasible. We've tested that on large intracerebral hemorrhages. But what really matters in clinical decision making is sensitive detection, and it's not there. And there are a variety of reasons why that's the case. In the setting of acute stroke, I doubt, I could be wrong, but I doubt that any MR will displace the non-contrast head CT, which is the first port of call for an acute stroke patient presented to the ED. Just it's so fast it's so embedded in workflows. Unless you are a patient presenting in France, where they do acute MRs in lieu of CT. It's the only place in the world that does it right now. And NIH was doing it for a while at Suburban Hospital. I don't know if they are now. Aside from those locations, it's CT, given its speed and sensitivity.
Everyone's getting a CT, that's kind of a given.
That's a given.
The hemorrhage question, it's kind of off the table for what we need to do.
Exactly. So then the first stop is a CT. If you are trying to rule out a TIA or a small stroke, then you have a little more time because you've already gotten your CT, you've excluded hemorrhage, and then you can consider it as a triaging tool. There are other scenarios in which I think it would be really advantageous for a low field MR scanner to detect hemorrhage. And I'll talk about that next, and that's in the setting of microhemorrhage, which I think is quite a bit out of reach right now, and then other forms of hemorrhage, such as like a subarachnoid hemorrhage or a cortical superficial siderosis, all of which can occur in the setting of lecanemab or anti-amyloid therapy, which is another use case that I'll transition to. But what I would say is for this audience is that I know Matt and I have had conversations about how to detect hemorrhage directly or indirectly. But if there is an innovative solution for detecting hemorrhage and microhemorrhage and chronic hemorrhage, it would be a game changer. Susceptibility weighted imaging and gradient echo imaging is not really feasible at low field for reasons that I'll let others explain, but it would be incredibly helpful clinically. Okay, so I'll move along because I know I want to be mindful of the time. So in parallel to the acute stroke work, one of the things that really came out of I still remember the meetings. I think of the three of us with Eugenio and Matt. They invited me into a conversation and where we were just brainstorming ideas. And Eugenio was sharing some of the amazing work that he and his team had been doing on clinical grade MR scans and using machine learning tools to reconstruct or super resolve them into 1 millimeter like scans. And very quickly it became clear that this could have powerful implications for the low image quality and low SNR of low field images. And that led to Eugenio and his group adapting his existing, now very widely used SynthSR algorithm to low field and training it to include synthetic images that recapitulated some of the contrast, resolution, and orientation characteristics of low field images. And wow, remarkably, it was able to create, you can see on the right-hand side, a low field image that was super resolved for all the world, looks like a T1 MP range, which could then be used for downstream tasks such as segmentation. And the initial method that Eugenio developed was to combine a T1 and T2 from the low field scan, which you can see on the right, process it through the low field version of SynthSR, and then it really looked remarkably similar to the 3T MP ranges. And in that initial work that Eugenio led, we were really encouraged by the correlations between the low field and the high field. And with that work, we then realized, okay, it's worth putting in the time and energy to really optimize both the image acquisition and then start enrolling and scanning patients where morphometry is useful from a clinical standpoint. And so we started with a healthy volunteer cohort, and we scanned them at low field over in the neurology outpatient clinic. And then here, I think it was Bay 4, the 3T scanner. And that gave us the ground truth and the low field images. And in parallel, Eugenio and his team fine-tuned the algorithm to different voxel sizes, was able to train it so that we only needed a single image input rather than two, as well as making it robust to image orientation. And the right-hand graph is a DICE score summary of these 25 healthy volunteers that we analyzed, looking at the spatial overlap on the segmentation masks that were derived from low fields compared to the ground truth at 3T. And what you can see is that version 2 led to higher DICE scores as compared to the initial version 1, and that both T1 and T2 were nearly equivalent. So that inspired us or motivated us to acquire low field images at different voxel resolutions and orientations. And the reason for this is that obviously we knew a 1 millimeter isotropic T1 acquisition on low field would be optimal for downstream segmentation tasks, but that takes too long. So we had to strike a balance in trade-offs between SNR resolution and then acquisition time. And so because we had healthy volunteers who were happy to sit in the scanner for extended periods of time, we were able to acquire all those images, super resolve them, and then segment and compare to ground truth. And here I'm just showing hippocampus. We've done this for a variety of other brain masks. And the bottom line, the key takeaway is that first, test retest on the right was really quite incredible. If we scanned a patient at low field and then on a separate day brought them in, scanned them again, processed both images through SynthSR and SynthSeg, that the DICE scores, which are what are shown here, were really quite high, suggesting excellent test retest. The other takeaway is that as long as the image acquisition resolution was isotropic and anywhere between 2 and 3 millimeter voxel size, that it was about optimal for accurate accuracy as here based on the DICE. We also looked at correlation coefficients and various other metrics. The advantage to this is that a 2 millimeter isotropic acquisition is like 10 minutes or 7 minutes, but a 3 millimeter is 4 minutes. So that was the game we were playing, is trying to get it good enough resolution, but short enough that it could be tolerated by dementia patients who might not want to spend time in the scanner. So once we had solved that, from an empirical standpoint, we then started scanning patients with mild cognitive impairment and AD who presented to the neurology clinic, and then partnering again with our colleagues at Yale. They had scanned some vascular dementia patients with vascular risk factors who had presented to the ED for non-neurologic reasons. And that gave us a cohort of vascular dementia, MCI, and AD. And all of about the same age. The vascular cohort was a touch younger, but not by much. So then we acquired the images. You can see the image resolution continues to improve, the native images coming off the scanner. And we were able, in this case, we focused on the hippocampus, the lateral ventricles, and the whole brain, because those are the most common metrics used in AD for morphometry anyway. And again, this is just a correlation scatter plot showing really nice correlation, even in patients with disease who had, in each case, all these patients had high field images that we used as ground truth. And so then we asked the next question is, since we knew the low field data was as reliable as the best case scenario scanning healthy volunteers, we next asked the question, can that low field information be used to compare different cohorts, which is what I'm showing here. And what you can see in orange on the left is that the hippocampal volumes of these patients corrected for intracranial volume, a standard method of assessing the size of the hippocampus, they were smaller in the AD and the MCI patients as compared to the vascular dementia patients. The lateral ventricles were also larger, and their whole brains were smaller, which is exactly what we were hoping to find, and we were reassured by that. And then finally, in the last couple minutes, I'll mention that, again, partnering with Eugenio and his team, we wanted to start to push towards automated quantification of pathologic lesions, not just morphometry, but recognizing that many patients that we see have pathology, of course. And here, Eugenio developed an algorithm to automatically segment periventricular white matter hyperintensities, which are an imaging marker of vascular and cerebrovascular disease. And so we applied that again at high and low field, and focusing on that vascular dementia cohort that I had just described. We again validated that whether you look at the correlation on the left or you compare it to a semi-quantitative assessment of white matter hyperintensity burden called the Fazekas score, that it was really quite nicely related to each other, where a higher WMH volume derived from Eugenio's algorithm was associated with a higher Fazekas score, is exactly as we would have hoped. So bring it all together, a combination of imaging dementia patients, focusing on specific parameters in the acquisition to make sure it's as tolerable to patients. And we can now derive WMH volumes as well as a variety of brain morphometry volumes. This summarizes it here. We can, in a cross-sectional analysis, we can show differences between Alzheimer's or beta amyloid-related pathology and vascular pathology. And I will say that we have now, because Bruce raised the question, I'll mention that we have now started scanning patients who are undergoing every other week anti-amyloid therapy, with the idea that perhaps this low-field scanner can be used for disease monitoring of pathology. And we've been able to capture both severe and mild ARIA-E cases. But as I alluded to earlier, ARIA-H, which is the hemorrhagic forms of complications of the therapy, are currently not within reach. So, in summary, the overarching goal of portable low-field MR is to improve access to MR, and I mean that in any environment. It means when a patient comes in and has an interaction with a healthcare provider, gosh, wouldn't it be nice just to get as much information in that one visit as possible, rather than fragmenting it out, which is a structural limitation of our healthcare system. So being able to improve access, making it patient-centered and more convenient, is really the value add that low-field MR can bring. It's safer, it's easier, it can be convenient to patients, but that is, with all things with MR, there are always trade-offs. And the trade-off here is that image quality is lower. There are a number of ways in which we're thinking about how to overcome that. And so the takeaway, at least from our experience, is that how to embed this into clinical workflows and defining use case scenarios that change practice requires careful and thoughtful integration into clinical decision-making and ultimately testing that. We're at the phase where we're not yet at the phase where we can prove that embedding it into clinical decision-making improves care, but we're getting close. And one of the most exciting things about this, as I've said already, is not only the multidisciplinary nature, but that it is so rapidly changing, it just makes it fun. And it makes it enjoyable. And again, I want to, I'll skip this, and I just want to thank you all for your attention. I want to acknowledge, again, the many team members that have participated in this project. This is not a comprehensive list either. And thank you for your attention.